Well, good afternoon. Thank you all for being here at this really important symposium. When I arrived 6.5 years ago, I was talking the language of One Health, and I was pleased to discover that our own College of Public Health was deep into this. Over the years, I want to acknowledge and thank Dean Hakim for all of her support of the idea of One Health, and this symposium is part of that. I also want to thank Professor Von Hippel for bringing his talents to Tucson to help lead this incredible effort. It's part of our strategic plan. It's also made its way into support from the legislature, and we're incredibly thankful for that support. I'd also like to, it's been a while now, but thank all the members of the Public Health Advisory Team that helped so much during the pandemic to help us work through the pandemic on campus. It's my honor and privilege today to introduce our keynote speaker, Dr. Peter Agre, who is the Bloomberg Distinguished Professor in Molecular Biology and Immunology at the School of Medicine at Johns Hopkins Bloomberg School of Public Health. We're looking for our own Mayor Bloomberg to help support our university. You're incredibly blessed to have him. Professor Agre was also the director of the Johns Hopkins Malaria Research Institute, and importantly, was the recipient of the 2003 Nobel Prize in Chemistry for his discovery of aquaporins. He's been a member of the U.S. National Academy of Sciences since 2000, and served as president of the American Association for the Advancement of Science from 2009 to 2010. We're incredibly fortunate. It's rare that you get to meet a Nobel Prize winner and have them impart their knowledge and wisdom to us. So we are incredibly fortunate to have you here today, Professor Agre, and we're looking forward to your talk on Aquaporin, water channels, and One Health. Now it's my honor to turn the podium over to you. Thank you for being here.
Thanks, Barry. Good afternoon. It's a pleasure to be here at the University of Arizona, where so much great science has gone on in the past and it's going on now. And One Health is clearly the medicine of the future. I'd like to thank Fred von Hippel and Griselda Ruiz-Brown for their gracious hospitality. You really know how to treat someone very generously. So I'm going to break my talk into two parts. One, the story of the aquaporins, which is pre-Google. This is kind of antique science, but sometimes it was productive. And then talk about malaria in Africa, an application of this basic information. So in the beginning, I had hair, articular knee cartilage. I was 21 years old, and I was leaving my home state of Minnesota to head to Johns Hopkins because they were very well known for their work in tropical medicine, we called it then. Now it's global health in the developing world. This is an Arctic river, the Thelon River. And like many streams, it flows gently. Water is a peaceful and ubiquitous part of biology and geography. But when it's at disequilibrium, it can create a tremendously dangerous torrent. And in our bodies, which are 2/3 water in mass, we have water circulating entirely quietly. During the time we're here together, we'll be filtering plasma into our primary urine to reduce toxic waste. We'll be secreting and reabsorbing cerebrospinal fluid. And all of that's occurring because there's a special plumbing system that exists. And we don't think about it until things go awry. Problems like brain edema, cystic fibrosis, renal failure. These are all diseases which are manifestations of aquaporin biology. So how did we discover aquaporins? By sheer blind luck, but with a little wisdom and help from many friends. It's often said it's better to be lucky than to be smart. I think my lecture may illustrate that it's even better to be lucky and have friends that are very smart, and they make you wise. The contributions of the others in the field were so important. So this is Brad Denker, when he was a postdoc fellow. Now he runs the nephrology program at Harvard Medical School, Brigham and Women's Hospital. But he noticed on our purifications of the rhesus blood group antigen, which was a biological first. Rh antigens cause problems with hemolytic anemia of the newborn. A second protein came along as a contaminant. Now, a wiser scientist would have got stuck to his specific aims, but I feel like I have a little bit of Huckleberry Finn in me. And sometimes the unexpected produces some irresistible curiosity. So this 28 kilodalton protein, which we probably should have disregarded, turned out to have some interesting features. For one thing, no one had ever seen it before because it doesn't stain with Kumasi blue, the typical stain which proteins are analyzed. These are polyacrylamide gels. But this very sensitive reagent, silver reagent, which is basically like a photographic development, shows that we could extract and purify amazingly abundant protein, which wasn't known to be there. So the brilliance of the discovery is in part like driving some remote desert area and coming upon a missile site or a city of 200,000 people that's not on the map. It gets your attention, something new here. And that was Barb Smith, who was our technical assistant for so many years. So without knowing what it did, and without asking NIH's permission, weekends and at night, we did a few experiments that turned out interesting, and we decided we'd clone it out. Now, we were cloning out a new protein because it was new and we thought it would be important. There was no greater hypothesis. So it was a bit of a risk. Had we not succeeded, I'm sure I would have faced unemployment when the grants ran out. But when you're lucky and you have friends that are smart, things turn out. And the complementary DNA which encodes the new protein shows that it's a membrane-spanning protein at the outer surface of our cells. And it has an internal repeat, meaning the first half of the protein and the second half of the protein are genetically related, not identical. Still didn't tell us what the protein did. So we went to the genetics database and found there were some related proteins in other tissues, other species. But none of them gave us a clue what the protein did. There was a protein from the lens of eye from beef cattle, a major intrinsic protein was referred to, a protein from the brains of insects called big brain, a protein expressed in bacteria and in plants, but none of them were functionally understood at a biophysical level. So we were still stuck. How do you solve a problem when you're stuck? Well, you can go to the library, but there are a lot of things to read in the library, and talking to other people turned out to be the pathway which led us to success. So the conversation that led me to think this protein might be the protein that allows the entry and release of water from cells was provided on a family camping trip to Florida. After making several vacations in national parks, which is a wonderful way to have a vacation, and on a basic science salary, that's the vacation we could afford, a tent. The kids decided, when we asked them which national park they'd like to go to next year, they decided they'd like to go to Disney World, which is not a national park. But families aren't true democracies, so we actually went to the Everglades, and they quickly realized those are real alligators. We then went to Disney World, and on the way back to Baltimore from Florida, we stopped in North Carolina, where I worked for 3 years at the University of North Carolina. And my clinical supervisor was John Parker, an eminent hematologist who had a small physiology lab. And I told him about this new protein. John leaned forward and said, you know, red cells, and it also exists in renal tubules, homologs in bacteria and plants. Have you considered this might be this long-sought water channel, which physiologists have been searching for a century? I had never heard those words juxtaposed, water and channel. It was John's suggestion. We teamed up and pursued this study that I'm going to tell you about. So all cells have a finite degree of water permeability, like their basement in wet areas of the country, probably not Tucson, but water seeps through. But some tissues have abundant movements of water. which can't be explained by simple diffusion, and they were speculated to contain endogenous water-selective channels. But the biophysicists couldn't identify them. They didn't know what it was. It was a mysterious process that made red cells and salivary gland epithelia and renal tubule epithelia permeable to water. And teaming up with Bill Gugino, this is some years later, we were both young scientists when we started working together, we tested water permeability. Now, how could you measure water permeability of cells? If you radioactively label water with tritium, you can't measure it because the protons hop between molecules. There's no photoaffinity method to label water. And so it was set upon expressing the new putative channels in frog eggs. Frogs lay eggs in freshwater ponds in the springtime, and the oocytes in the fresh water are resistant to water entry. So we figured if we could express a water channel in a frog egg, it should swell and explode. And that's exactly what happened. On the left, a test oocyte. On the right, an oocyte, a frog egg, about 1 millimeter in diameter, injected a small amount of complementary RNA. And when it transferred to distilled water, it exploded like popcorn, producing much jubilation in the lab. I think one of the reasons people pay basic science such low salaries is because they get such great joy from discovery, they'll do it anyway. And this is Greg Preston, to whom we owe a great deal of credit. He's had a long career at Pfizer Pharmaceuticals and been a very successful colleague. So just to confirm it, since we could purify reconstituted membranes, these are small synthetic vesicles, and found that the ones that contained the water channel protein would shrink or swell with millisecond kinetics. This is a stop flow analysis, but it shows that the movement of water is almost instantaneous. But the background movement of water is not zero, so it's a matter of increasing the velocity of water. And we came up with a putative structure to merge the primary sequence, figuring that it folded together to form what we refer to as an hourglass, a protein that spans the membrane and allows the outer water from the outside of the cell and the inside of the cell to meet at a very narrow aperture. And this was confirmed by structural biology studies with Yoshinori Fujiyoshi and Andreas Engel and their team in Kyoto and Basel, Switzerland. And shown here is a cross-section. Now, this is already 24 years ago. Time moves so fast. A cross-section of the protein viewed from above shows a donut with a single pore in the center of 3 angstroms in diameter. And the cross-section on the right shows that the channel moves from side to side. 3 angstroms is precisely big enough for water to move through it, water being the smallest biological molecule. But there were barriers to the movement of other solutes and sugars. So it's a very nice, tight fit. Water fits through and moves. And these structures were confirmed in aquaporins from diverse species: bacteria, plant, microbe, vertebrate. They all have these, and so every living organism has aquaporin, or a related protein. This is work of David Cosono, who's now assistant professor at Harvard Medical School. He was a student at the time when he worked with us. So water gets through, and it's very selective. But where does it function? Real estate agents are famous for telling you the importance of a house. It's based on the location, and the location. And with Søren Nielsen, a young Danish research scientist, we teamed up and mapped out the distribution of the aquaporin protein in kidney, which is, of course, very important. Our kidneys will filter about 200 liters of plasma every day. It's the same plasma being filtered again and again. So the primary urine is large, 200 liters. That's a 50-gallon drum of fluid. If we didn't reabsorb the water very efficiently, we would die of dehydration. And in green, I've outlined in the nephron. This is the primary functional unit of our kidneys. You'll see that the protein is expressed at the proximal nephron, where water transport is constitutively active. And the staining shows it exists in the tubules, both the lumen and the intraepithelial membrane. It doesn't exist in the collecting duct, which is the terminal part of our kidneys where vasopressin acts. So the protein is abundant. This is an immunogold decoration with gold particles labeled labeling antibodies. And the surface of these epithelial cells have a hair-like projections where they're very efficient in moving water, like having a mop with a large surface area. It allows water to enter the top of the cell, pass through the cytoplasm, and exit the lateral and the basal membranes. So every day, 200 liters of primary urine is being released, and probably 199 liters are reabsorbed. So what does this have to do with human disease? As a medical doctor, that seemed important, and we imagined some dastardly syndrome with renal failure and hemolysis and bad consequences. But together with Chulso Moon, we localized structural gene for the Aquaporin 1 protein at the short arm of human chromosome 7. This was before the genome had been solved, so you know this is ancient history. Most of you young people were at least a decade before your birth. But at that time, there was a lot of information about localization of blood group antigens, and to have a coincidental location of the Colton blood group with a structural gene for our protein was a strong clue that they were one and the same molecule. And that turned out to be the case. And so we were able to identify of the world's Colton null individuals, there are only 6 known in the whole world. We were able to get 2 of them to come to Johns Hopkins for clinical testing. And Landon King, the young man with the necktie here, who was a postdoctoral fellow, did the phenotyping, which established the importance. Normal people sleep 8 hours or so, and awaken in the morning, they've not had fluid for 8 hours, and they're a little bit dehydrated. That first hit of orange juice tastes so good because we're dry. And our kidneys will concentrate urine, so we have a smaller volume of urine released at the end of the sleeping period. Individuals with nephrogenic diabetes insipidus release dilute urine, and they can't concentrate it. And the fact is, Landon established that the Colton null people are Aquaporin 1 null, and they can't concentrate their urine fully. But they can get through the night all right, but any further thirsting will lead to disastrous dehydration consequences. Landon, by the way, now is the executive dean of Johns Hopkins Medicine, a very important role at our university. And he's a pulmonologist, and what he discovered is that in our lungs, the expression of Aquaporin 1 is important at the time of birth when fluid moves from our interstitial, in our alveolar spaces back into the lung parenchyma to keep our airways dry. We were joined by several dozen laboratories throughout the US, Europe, and Japan who thought the aquaporins would be a good thing to study because they've been looking for explanations for various physiological phenomena. And I'm pleased to say now that I think the count is complete. There are 13 homolog genes to the Aquaporin 1 in human genome, but there are hundreds that have been identified in all other species of life. The Aquaporin 2 is the one that is most important clinically. The medical students all want to know this because this is going to be on their board exam. Aquaporin 2, like Aquaporin 1, is permeated by water, but it's regulated. It resides within the epithelial primary principal cells in the collecting duct, the last kind of wide tubule which allows the final concentration or release of urine. And the Aquaporin 2 is co-localized with Soren Nielsen in collaboration with Mark Knepper at the National Institutes of Health. And it was Mark who first coined the term aquaporin to describe this protein. So unlike Aquaporin 1, which is constitutively present at the surface, Aquaporin 2 resides in intracellular vesicles. And the electron micrographs on the left may be hard for you to see, but the diagram on the right shows an intracellular vesicle, which in response to vasopressin, that's the antidiuretic hormone released from our pituitary glands when we become thirsted. Aquaporin 2 then traffics to the cell surface, and it allows water to pass through the primary epithelia. So this is the antidiuretic state. When we are thirsted, out playing tennis a hot afternoon in Tucson, we can't keep up our hydration. We become fluid deprived, and our kidneys will maximize the reabsorption of water. The same individual later in the day getting hydrated and having a beer will face the opposite. Now we have fluid excess, and the alcohol in the beer will inhibit the release of vasopressin. So that leads to the diuretic state, large volumes of dilute urine. And we go back and forth between these two states constantly. Desert rodents, probably in the Sonoran Desert here, certainly in the Joshua Tree, those desert pack rats are able to concentrate their urine to 6 molar. We can max out at about 1,000 millismolar. They're at 6 times the concentration ability because of the expression of the aquaporins in their elongated collecting ducts. So it explains a lot about biology. Another homolog, the aquaporin 0, is also referred to as MIP, major intrinsic protein, and defects in this result in cataracts in small children. Cataracts are very common in the elderly. I'm developing cataracts, I know, because my ophthalmologist is aware of that. But small children are usually not affected. Fortunately, the removal of the cataract, sight is restored, but it shows that the importance of the protein has diverse phenomena based on the distribution of the homolog. The eye is a complicated organ, probably as complicated as the kidney. So laboratories around the world led to the expression and identification of several homologs of the aquaporins, including work here done at the University of Arizona by Dan Stamer when he was here working with Robert Schneider and John Regan. They established the distribution of the aquaporin 1 in the anterior chamber of the eye where aqueous humor is reabsorbed. Dan is now a faculty member down at Duke. In brain, we have a special problem. If you twist your ankle while playing soccer or tennis or jogging, we know what will happen. No major damage to the tendons and muscles may occur, but capillaries may be ruptured and the swelling will occur. So we're put on crutches for a couple of weeks. The swelling dissipates and there's really no harm. But if someone sustains a brain injury, it can be very difficult because our brains are encased by our crania, which are rigid, to protect the brain. But if damage occurs, it will at least result in swelling, which will lead to compression of adjacent brain tissue. And this can lead to a fatal outcome. We teamed up with Ole Petter Ottersen. This is when he was a young Norwegian neuroscientist. He's now just finished as a rector of Karolinska Institute in Stockholm, and localized the aquaporin 4, the brain homolog, at the outer surfaces of the brain in the glia limitans and inside the brain parenchyma surrounding the brain capillaries, the so-called blood-brain barrier, which keeps our brains free of toxins and molecules that can't pass across the blood-brain barrier. But this is a site where much damage occurs from a head injury. I'll skip some of these data slides because they're going to be very complicated. But the protein lives in its native sites in high density and forms membrane crystals. It's amazing. Who would think that membrane proteins floating around freely would associate with each other and make almost a fabric-like assembly? But they have enormous ability to move water. An experiment done by our colleagues in Norway, led by a Iranian-born neuroscientist, Mahmoud Amiri, shows the paradoxical outcome of a definitive brain injury in a mouse model. It was a temporary occlusion of the temporal artery. That's the artery wraps around the outside of our brain, and is the most commonly infarcted artery in stroke. So they were temporarily occluded and released, and reperfusion occurred, and the normal mouse sustained a fair degree of brain damage. You can see that the animals were sacrificed, stained with a vital stain. The preserved brain is red and pink. The infarcted and the edematous brain is white, and you can see that the normal mouse does much worse than the mouse that has mutations in aquaporin 4 or the structural proteins that hold it in the right place. This is interesting because usually mutations cause problems, not benefits. We know that the mice who have mutations in the structural proteins that are in the aquaporin 4 are vulnerable to epileptic seizures. But if they should sustain a brain injury, they'll have some benefit from the reduction in the edema. So the search for inhibitors for the Aquaporin 4 have been underway, and there's some that are being tested. I'm working with a group from Yale and Case Western, but it doesn't look promising that we have good medicines for Aquaporin 4. And the problems of brain edema are ubiquitous. I dare say everybody in this room has a friend or a relative that may have sustained a concussion, stroke, or a brain tumor, and the brain edema is horrific. This MRI of a middle-aged lady shows a large frontal lobe meningioma, a non-malignant tumor. And notice how large it's grown over probably decades. And it started to cause problems, headaches, and loss of appetite. And when the physician sent the individual for a CT scan, this humongous tumor was obvious. Now, the MRI in the upper right shows the MRI weighted for the density of water. And the problem is caused not by the tumor itself, but the water that is surrounding it in the brain edema. Surgical removal, shown in the lower right, the tumor's gone, the edema took some time to subside, but the life was saved. It's important that not only in response to injury and tumors and infarcts, the movement of fluid through the brain during sleep. This is work of Make and Nedergaard from the University of Copenhagen and Rochester University. She has a hypothesis that the sleep movement of water through the brain, carried by the Aquaporin 4, is necessary for brain homeostasis. And the inhibition of this is the final pathway in many forms of dementia. It's a hypothetical study at this point. It looks very promising. I'm going to skip quickly through some of the other information. A salivary gland, aquaporin 5, abundant in salivary glands, sweat glands, lacrimal glands, the last membrane where water crosses during the generation of those fluids. In mice that have been disrupted, the aquaporin 5 gene has been disrupted, have sweat glands, but they don't release fluid. Now, desert rodents concentrate their urine. They don't need to sweat as much because they're out of the sun during the day and hiding in the cool shadows of the Joshua trees or wherever. Humans work in the hot sun, and the ability to release sweat is essential for survival. So there's another part of the family, the aquaglyceroporins, where the pore size is slightly different. Glycerol, a 3-carbon polyol, is a substance we're all familiar with, but I'd never thought about it much in terms of clinical medicine. But it's quite clear that the bacterial homologue, known as GLP-F, is the way glycerol is taken up by bacteria. It's in the human protein originally called aquaporin 3, we call it aquaglyceroporin 3. It's abundant in skin and in the basal levels. Now, we were visited in our laboratory some years back by a group of the executive officers from the Christian Dior company. I don't know about you, but we don't get many visitors like that. And I was a little suspicious of their motivation. And as I expected, had a commercial product that was coming on the market, Hydraction skin cream, which their chemists had identified some small molecules lead to increased expression of the aquaglyceroporin 3 in sun-exposed skin. Now, the beauty industry has to show that it's not dangerous. They don't have to show that it makes you beautiful, even though their advertising would suggest that. It was very expensive, I think for 50 euros for a 50-gram jar. And this is the back cover of the Marie Claire beauty magazine, which I purloined from the Charles de Gaulle airport. And in French, it talks about spectacular results, hydration, profound hydration. In the bottom, the Nobel Prize in Chemistry. I showed this to my mother back in Minnesota. She was 85 years old at the time. She smiled. She said, "Peter, I think you're finally doing something useful." Imagine that. Our mothers know us better than we know ourselves. So it's always been a quest to do something useful, and I hope we've done something useful. So I'll just close my scientific presentation and shift to the malaria side. Two other glyceroporins are present, one in fat and one in liver, which allows us to store excess calories as fat and release glycerol during fasting and starvation. The one human mutant with the Aquaporin 7 is perfectly normal, but he can't release glycerol after profound exercise. He could lead a normal life, but he could never finish the Tokyo Marathon. Now, that may not seem important, but during human evolution, those who could exercise beyond the capacity of others would have a survival advantage. And the glycerol is taken up by the liver where it's turned back into glucose. So the late stages of starvation, this probably is a very important pathway. And this is work of Jen Carberry, who's now a faculty member at Duke. Aquaglyceroporins are also the transporters of certain heavy metals, notably arsenite. The United States has had some problems with groundwater contamination. In Eastern India and Bangladesh, there's a natural presence of large amounts of arsenite. And the surface water is contaminated with cholera, so the people are faced with a very grim situation. And there was so much arsenite ingestion that until a couple of decades ago, there were something like 40,000 cases of hepatocellular carcinoma showing up every year because of the amount of arsenic that was taken up. One other member of the family I'll mention just briefly was a big surprise to us because we did a series of experiments showing that Aquaporin 1 was permeated by water exclusively and no charged molecules could go through. When we studied the Aquaporin 6 from the renal collecting duct intercalated cells, we found that all the staining and intracellular vesicles alongside the proton ATPase. This is how our kidneys would respond to acid or alkali. And there was an ion conductance which we characterized. And I thought back about a study published years before by Andrea Ewell, Dan Stamer, and John Regan here at the University of Arizona, where they found an ion channel behavior in Aquaporin 1, and none of us could reproduce it, so it led to a little bit of a brouhaha in the literature. But they stuck with it, and they confirmed that Aquaporin 1, under special situations, can create a gated ion channel. It's not altogether clear what the biological significance is. It may not exist in all tissues, but it's a new wrinkle. And it shows you what this is ascribed to Mark Twain. What gets us into trouble ain't what we don't know. It's what we know for sure that it just ain't so. And so I stand corrected. I think science corrects itself. That's one thing about our work. The bacteria also contain a glycerol and a water channel, Aquaporin Z and GLP-F, work of Giuseppe Calamita, now a professor in Italy, Mario Borgna at the North Carolina Electron Microscopy Corps. He's the director. Yeast also have aquaporins, but this was an interesting lesson because while yeast have two glycerol transporting homologs and two water channel homologs, laboratory strains of yeast have the genes, but they all have mutations. So it shows you don't have to have aquaporins functional to be alive. Lab strains of yeast are alive, but they're probably selected because the aquaporins lead to damage during the washing stages of laboratory cultivation. So there's a surprise that occurred in my life. This is 20 years ago last fall when the phone rang early in the morning. A pleasant voice on the other end. This is an important telephone call from Stockholm for Professor Peter Agre. Are you Professor Agre? I answered, I sure am. And he informed me I would share the Nobel Prize in Chemistry with Rod McKinnon, who studied ion channels. He was at Rockefeller University. And by the time I got to the laboratory, our team was already into their second or third bottle of champagne. The university president came over to see me. President Robbins, I hope you have that occasion. But I had to ask myself, until that moment, I didn't realize we were such close personal friends. Donors like it. So this was the young people in the lab. I went to my office and the voicemail was already filled up. And I came to a familiar voice with a Norwegian accent. Peter, we've just heard the news. It's unbelievable. And then there was a pause and the voice came back on. No, it's believable. We believe it. even the neighborhood cut-rate liquor store got into the celebration. And I'd like to defend my reputation. I am not their best customer. And here I am with my family. For you, may I pause? I have Parkinson's, and sometimes it freezes up. So they're the support crew that made all this work. And I think that's something that scientists share, their friends and their family, that keep them going. And really, from maybe perhaps some cynical view, the Nobel Prize is a wonderful thing. You get a lot of attention, but it's really a reward for the whole field. And in October, first week of October, every year on public radio at least, many of the commercial stations, the first thing they'll announce is the Nobel Prize in Medicine on Monday, Physics on Tuesday, Chemistry on Wednesday, and then they vary it, Literature, and finally Peace. And it's, I think, a great public relations for the science in many laboratories, because we're all after the same objective: to make discoveries which improve life on the planet. So I was faced with a decision before the Nobel of moving into new areas with the aquaporins, and I always wanted to work on malaria. When I came to Johns Hopkins, that was my objective. I worked on cholera for a while as a medical student. Cholera, of course, is a horrible diarrheal disease. And I was really caught up in the work. I remember going to a mixer at Goucher College, meeting a young lady. She seemed very nice. And she asked me, Peter, what medical specialty are you interested in? And I said, diarrhea. That was the end of that. And then I met Mary, who was sitting in the first row. Farm girl who grew up outside of Ellicott City, Maryland, and she was working in Diane Griffin's neurovirology laboratory at Johns Hopkins. And she wasn't squeamish, although we fell in love, we got married, and she did establish a few things. She said, Peter, if you're going to work on diarrheal diseases, please don't bring your work home at night. Probably a wise way of proceeding. Well, it turns out malaria parasites also have aquaporins. There are two different genes. Eric Beitz, who was at Göttingen and now is at the University of Kiel, spent 2 years with us as a habilitation scientist, and Slavica Juranovic from war-torn Serbia worked on the aquaporins aquaglyceroporins from malaria. And they found several interesting things. First of all, red cells also have the aquaglyceroporin 3. And together, when the red cells are invaded by a malaria parasite, this forms the pathway which glycerol from the plasma can move across the red cell membrane, the parasite's membrane, and be chemically re-engineered into phospholipids. And the parasite will grow and divide. In the very center left, you see a ring stage, a single malaria myriazoid invading a red cell. And in the far right, lower third, you can see it's called a schizon. Those parasites over the next day and a half will divide up to 32 daughter cells. So we have a logarithmic expansion of the infection. First one parasite, 32 squared is 1,000. 1,000 squared is 1,000,000. Pretty soon there are billions of these parasites circulating in the bloodstream of the infected individual. And it's a horrible pandemic which has been going on for thousands of years until the 20th century. Malaria was prevalent in Northern Europe, throughout the United States, much of Asia. And I'll skip the details of the function of this to get onto the malaria story. So about 20 years ago at the millennium, there was a discussion worldwide, could we actually eliminate malaria? Because 20 years ago, about 250 million people were getting malaria every year, and 1 million children were dying. More than 90% of the malaria deaths are children, age 5 or younger, and most of those were in sub-Saharan Africa. Sub-Saharan Africa has had difficulty with malaria, which probably originated in a transmission from gorilla or chimpanzee sources at the dawn of time. The weather in sub-Saharan Africa has two basic seasons, the dry season and the wet season. And the countries I'm going to describe for you, the dry season extends from late October till April. From April to late October. So our summer is their dry winter. And then from late October to April, it's the wet season. So they're in the southern hemisphere. And so the countryside turns from a parched desert, maybe not too different from the northern Sonoran Desert, to with the appearance of the wet season, things start to grow again and life becomes very lush. Plants contain aquaporins and they're different tissues. They're actually more complicated than we are. But one member of the aquaporin family in the rootlets explains how water can be taken up so efficiently. So this is work of Ralph Kaldenhoff from the University of Würzburg in Germany. Arabidopsis is a small mustard plant and a favorite experimental subject for plant physiologists. So the wild type, the parental strain on the right, you can see that the lush arboration with arbor formed from a rather modest amount of rootlets. On the left, you see a plant which Ralph has introduced a gene disruption so that the aquaporin expression in rootlets is reduced by 90%, so only 10% as much water absorption capacity. But the plant is very savvy, sends out more rootlets to compensate. Plants compete for water. I'm sure you in Tucson know that very well. Mosquitoes also have aquaporins, and it's a bit complex. This is work of Connie Liu now at the FDA, Jason Raskin now at Penn State University when they were young scientists in our lab. And I'll tell you just about one of them, the aquaglycerin 3, which is expressed in several tissues. And by introduction of small inhibitory RNAs, we were able to make mosquitoes that were deficient in the aquaglycerin 3. And the print is pretty small, you can't see it, but on the right panel B, there are 3 different tracings. And these are survival curves, how long the mosquitoes were incubated at 39 degrees centigrade. That's about 102 Fahrenheit. And the tracings show that the mosquitoes with a reduced amount of aquaglycerin 3 survive less. They succumb sooner, so it's a protective molecule for the insects to survive the hot part of the summer. It's also a site for the oocytes, having a hard time with my words, when the gametocytes from a blood meal taken from humans is ingested by the mosquito, they will combine and form a zygote which invades the midgut, and the fluorescent markers show a method for identifying how many oocysts occur after a mosquito is taken in a blood meal. It shows that the aquaglycerin 3 is probably a receptor for parasite in the mosquito gut, which may be another drug target. So I'd like to give a little history of this part of the world because it's so fascinating and such a beautiful place. Zambia, where most of our work is ongoing, and this is funded by both the National Institutes of Health, the International Center of Excellence for Malaria Research, as well as with Bloomberg Philanthropies. It's a beautiful country, but they have a lot of hardship in the countryside because of infectious diseases killing their children. Now, the children of rural Africa have a life that's quite different from children in this country. They seem to be able to entertain themselves with no electronic toys. And when they're well fed and taken care of, and they go to school and prevented from malaria, they have wonderful lives. These little fellows were playing outside of our main research station in Macha in southern Zambia. The hospital there was a mission hospital. The faith-based community has made a lot of contributions to healthcare in Africa. This is Macha Hospital, built by Philip Tuma's father, Alvin Tuma. And Philip Tuma, who I'll show you in a moment, shown on the left here, is Pennsylvania born, but he grew up in Zambia and Zimbabwe. Back then they were called Northern Rhodesia and Southern Rhodesia. Here he is alongside Chief Macha. Among the regulatory steps to do research in Africa, of course, we have our own National Institutes of Health standard, and our Johns Hopkins University Review Board, but the Zambian government has their review board, and then you have to sell it to the community. In a tribal society, the chief is the spokesman, and the chief and his headsmen make decisions. Well, Chief Macho is a University of Zambia graduate. He's a farmer, and he's very, very wise. And Philip Tuma and Chief Macho decided 24 years ago that they wanted to launch an effort to eliminate malaria from the countryside. And Phil, of course, he grew up in Africa. He came back to the U.S. for medical school. He became a chief resident in pediatrics at Johns Hopkins, and he's returned to Africa until his retirement. Recently retired and returned to the States. And here's a little boy who's recovered from malaria. It's a treatable disease. It's better to prevent it, but everyone doesn't survive. So of the 600,000 children that die of malaria, there are probably several million who are infected. They may survive, but they never regain all of their normal functions. This child was stricken with cerebral malaria, a particularly deadly complication, and his life was saved, but you notice the disconjugate gaze, the cerebral edema from the malaria has damaged his visual cortex. So you can imagine the kind of handicap being blind in a third world country would be. And here is an experimental model generated from our laboratory showing that Aquaporin 4 is part of the pathway of defending against brain edema from cerebral malaria. So our institute had generous support, wonderful support from the Bloomberg philanthropies and Mike Bloomberg himself. And we were able to build a small research campus in southern Zambia and recruited these young scientists from the University of Zambia and the University of Zimbabwe. We were working very hard to do the participatory and epidemiology that we heard about. This is a pandemic that is not going away fast, but they've been remarkably successful in following up cases. When a child is taken to a rural health clinic and they confirm a malaria diagnosis, then the surveillance team goes out and to the countryside, first by a motorbike to localize it, and then with satellite tracking to have a research team test everybody in the neighborhood for malaria. And that was underway, and the introduction of the artemisinin in combination with another agent. This was the miracle drug, which 2UU received the Nobel in 2015. And it was not being widely distributed by the pharmaceutical companies because malaria is a disease of poverty, but it was Phil who convinced Emeritus to test it in Africa, and so Zambia became the first African country to employ the artemisinins. And this is a 25-year survey of the disease burden, and the first 5 years, we had about 1,500 children admitted to Macha Hospital each year, and an average of 60 deaths So in 2004, you'll see, if you can read the numbers, that's the sixth cluster from the left. The number of cases dropped significantly because artemisins were used to treat all infected people so they couldn't infect other people. In 2005, the number of cases dropped substantially. There were only 6 deaths, down from an average of 60 deaths. In 2006, there was a drug outage due to improper filing of reports. The medicine was not available. And the death rate rose again to 24. And in 2007, the artemisinin as a combination were reemployed and have been employed ever since. An insecticide-treated bed nets. Many of these provided by the U.S. President's Malaria Initiative and by the different faith-based communities. And as you see, from 2008 onward, the death rate has been very much reduced. And from 2018, the last statistics on this graph is from 2020. There have been no deaths, but the parasite is still there. And we know it'll happen if the tracking does not continue. The pandemic will reappear. And doing this surveillance is not altogether trivial. The rainy season in sub-Saharan Africa, the people who are affected are in the rural outback. So getting to their domicile is challenging, particularly well into the rainy season when the grass grows a meter or even 2 meters high. But they're in the rural countryside are the farm families that are raising maize and peanuts and a few other crops and are surviving on $2 a day or so. These are amongst the poorest people in the world. But the surveillance has been useful. And here's Harry Hamampumbo, one of our team members from Macha. He's a local. He didn't go to the University of Zambia. He's a high school graduate, but very intelligent, and he's leading a team of surveillance to see which of the relatives and neighbors are carriers of the parasite. And the families themselves are very pleasant, but they're not used to the culture of Western medicine, but they're very grateful. They lead very simple lives using farm implements which were discarded in the Western world 150 years ago. And everybody works. The older children care for the younger children. They carry the maize to the marketplace. They have it ground into cornmeal for their major diet source. And the kids, they know how to have fun. I think the notion that we sometimes hear from politicians that they don't respect life in the developing world, this is hardly the case. I think that the children are the most precious commodity that the families will ever have. The resources go into the future of their children. I love this picture. The little girl, her school book upside down says, "School is cool." Maybe she's a future University of Arizona student. So Zambia left the colonial history in 1965 when the Brits gave them independence and Kenneth Kaunda was elected president. And they've had a society that has faced many challenges, but it's a wonderful society. These are the ladies of Mancha waving goodbye to us. Beautiful colored skirts. Zimbabwe, which was southern Rhodesia, the sister state of Zambia, has had a less fortunate history. In 1965, when the Confederation of Rhodesia and Nyasaland dissolved and Zambia became independent, southern Rhodesia was run by a white supremacist society led by Ian Smith. They named their country Rhodesia, and it was a brutal 15-year civil war. It's a beautiful part of Africa. The Zambezi River here separates Zimbabwe and Zambia. Zim and Zam, as the natives refer to them. But the liberation of Rhodesia was led by Robert Gabriel Mugabe, a Jesuit schoolteacher. But Mugabe ran the country with benevolence for about a decade or so, but things then just deteriorated. His wife died and he remarried, and suddenly there was a new urgency to support the regime, and regimes can perpetuate themselves by the actions of cronies who require bribes, and so the confiscation of farms, and everything went bad for 15 years until the Civil War ended. As I said, the Mugabe regime took over. And for the last 15 years of Mugabe's life as president of Zimbabwe, as the country is now known, it was very difficult. It was basically a police state. So how can you run a research operation in a situation where everything is controlled by the president, who's, of course, the wealthiest person in the country and plans to stay around for life? Well, I think sometimes a little Machiavellian approach is necessary. To be able to work there, we were able to employ this wonderful lady, Shungyu Munati, who's a University of Zimbabwe graduate and is an administrator for the not-for-profit agency called Biomedical Research Training Institute. It turns out, of course, she is Robert Mugabe's niece. So we have to make a decision. Are we supporting a cruel regime, or are we doing what's necessary to do the work? And it's an ethical decision. It's just not always easy, but we opted to do the work. Things have been taken care of. Zimbabwe is a gorgeous country. This is a tea estate in the Eastern Highlands. And there are jobs. This little fellow is having a great time driving the tractor for the tea estate. It's rich farmland, but they lead simple lives, dependent, basically like summer camp would be for American kids. They're dependent on the water for religious purposes, for washing, for drinking, for cooking. And the resources, as in Zambia and elsewhere in sub-Saharan Africa, the resources go to the education of the children. This gentleman, a grandfather, probably about my age, I talked to him. His name, he said, was Never. I thought that was a pretty unusual name. In Zimbabwe, often people will name their child Pray More, Give More, Love More. And I asked him, how did you get the name Never? He said, well, he was named Nevermore. He was the 9th and last child. Well, Nevermore has his dream is that malaria will end, so his children and grandchildren don't have to worry about it. Something we all can agree upon. But in the end stages of the Mugabe regime, the healthcare suffered badly because of the economy. It was the worst inflation in the world, and malaria reappeared, and every morning during the rainy season, mothers bring their children or grandchildren in with malarial fevers. They have very dedicated nursing staff. This group of nurses work 24/7, 365 days a year. But what limits them is their ability to get the necessary medicines. And his is Sungano, our colleague, who was a very bright school kid from rural Zimbabwe. He went to the University of Zimbabwe and by a miracle got a scholarship to study at Oxford in England, where he did his PhD and a postdoctoral fellowship. And rather than joining a European pharmaceutical company. He returned to his home where he's now the Dean of Life Sciences at a university in eastern Zimbabwe. And he plans to lead the eradication of malaria in Zimbabwe. Going backwards. The third country I just mentioned briefly, the Democratic Republic of the Congo. Somehow every country I've been to that's called the Democratic Republic doesn't seem at all democratic. Congo's had the most tragic history with when the Belgians left in 1960, it was in disarray. Patrice Lumumba led for less than a year, and he was assassinated, and it led to the regime of Mobutu Sese Seko, which bankrupted the country and led to the revolt and breakaway independence of Katanga province. So Congo is an interesting country. It's circumscribed by the Congo River, but it has a history of universities and culture that is not often recognized. This is Bill Moss, our epidemiology director at Johns Hopkins, lecturing to the Congolese medical students. Congo has enormous mineral wealth, potential wealth. It's estimated that there are $75 trillion of precious metals and gems that are in Congo. With a population of about 75 million, that would mean every man, woman, and child should be a millionaire. Yet they have amongst the lowest per capita gross domestic product in the world. And where there's been preservation of the colonial architecture, it's evident, but Katanga province is still very valuable because of the mineral wealth. But the countryside does not have paved roads. It's difficult to get there. The missionary pilots that fly up the medicines, and I'm pleased to say that the U.S. has been a very generous supporter of the distribution of bed nets and medicines for the people in these regions. And here Bill and I are with the Congolese counterparts at Kilwa village in eastern Katanga province. They have all the best wishes, but they're limited in getting dependable supplies. And the children shown here, wonderful kids, but most of them will get malaria, many of them every year. And it's always been a place of some instability. This is the monument at the place where Dag Hammarskjöld's plane that went down during the Congo independence revolution, and the revolution continued. Probably been more than 5 million deaths in Congo over the past 20 years from the revolts. So anyway, I've come to the end of my hour and sharing my view as an old guy that's been around for a while, looking back. Science was wonderful, very enjoyable, but I think the people that we met along the way were the biggest reward. And it's the sort of thing that makes it feel it was all worthwhile. So I'd like to challenge the young scientists here to stick with your studies. It's not a quick road to riches, but discoveries get made, and you never know. My advice is if the phone rings early in October, or early in the morning in October, answer it. You might hear a Swedish voice. So a little wisdom. In Mandarin, they have their word for crisis is weiji. Wei, time of danger. Ji, a time of opportunity. And I think that's what we all have to thank medical science for. Thank you so much for the privilege of speaking at your university. It's a delight to be here. I'm about out of voice, but I can do sign language. the very back. Over. Are you ready?
I have a microphone. Thank you so much. And I was very interested in, I first learned of your work when I was doing my research here at the University of Arizona for my doctoral thesis on vasopressin in the brain, and discovered the impact of vasopressin on brain function. And so I was quite interested in some of your findings on, and what you thought about regulation of aquaporins and how they might impact brain function.
Sure. Well, first off, the work on vasopressin was not from our laboratory, but from colleagues. There are a number of groups around the world that have worked on it. And in addition to being the antidiuretic hormone, its name vasopressin suggests vascular constriction. It also causes veins to constrict to reduce the amount of blood loss after injury, and probably has many functions in the brain which are more difficult to describe. Certainly in the, I'm blocking, this is a problem with Parkinson's, you run out of dopamine, you want to say something, you can't think of the word. But the vasopressin secretory neurons have an abundance of the Aquaporin 1 at that location. So they're clearly part of the pathway, and vasopressin regulates some of the aquaporins, but not necessarily all of them. I can't honestly be regarded as an expert on this part of biology, but I think it's a very interesting thought process that you're showing. So sorry I can't do better with that. I think there's a question over here, please.
I really enjoyed your talk, and I'll say I'm a neurologist who studies Toxoplasma gondii, who's related to malaria. But I'm actually interested in the Aquaporin 4, which I think you skipped over. That one's like just in terms of your thoughts about why NMO or de Vic's disease ends up causing, it looks like there's a good reason for optic neuritis, but the spinal cord, right? Those are the two areas that most often are affected.
I'd fish back the slide, but it's take a while. So the question has to do with neuromyelitis optica, which was in the past sometimes confused with multiple sclerosis. They're apparently clearly different neurological problems, but the Aquaporin 4 turns out to be the binding site for antibodies which appear in the peripheral circulation of individuals when they have a flare of neuromyelitis optica. And I believe they've now been able to confirm this by transferring the immunoglobulin from human NMO patients to experimental animals, but for a long time they couldn't confirm it. There's apparently approval of a monoclonal antibody now to prevent the flare by blocking the antibodies against Aquaporin 4, which they believe are producing the syndrome and not byproducts. So to explain the difference why in the retina on the optic nerve and along the spinal cord, I think it's pretty well agreed upon it. The appearance of the antibody certainly marks the disease and probably is a pathological antibody. But I think there's a fair bit of disagreement in that field. Do you know more about it than I do, probably? You ask tough questions.
So you mentioned talking about the president's malaria initiative and different opportunities in the growing field of health diplomacy and global health. I was wondering, how do you think these fields will change, especially in the age of globalization going forward?
Well, it'd be nice, since we have capacity to take care of some of these diseases, we could do a little better job. But it becomes complicated when you're dealing with other countries. We have enough trouble taking care of all the problems in our own country. But the President's Malaria Initiative, which is something launched by President George W. Bush, has been received bipartisan support and ever after has. It's been remarkably effective, and it's probably saved millions of lives. It has the other President's initiative, the PEPFAR, President's Emergency Plan for AIDS Relief, similarly provides resources for the countries in Africa and elsewhere that are most stricken by this. So in terms of our responsibilities during the age of globalization, it's quite clear that infectious problems like Ebola, SARS, COVID, they don't respect borders. And so it's not only our moral obligation, I feel, it's a practical obligation to have hypervigilant public health officials educated at programs like the Zuckerman School of Public Health here at the University of Arizona take on these jobs. And I think if we could organize better and stop spending so much time looking at military problems, we could take care of many of these issues much better than we do. So the resistance to medications like the artemisinin are now becoming worrisome. But given the current information, more vigilant and careful administration of the medicines will be effective for quite a few years still. But new medicines are being developed, and it has to be a priority. It seems like it is a priority when other things are not bothering us. But it's unfortunate that sometimes the public is distracted with other issues, and these problems affecting impoverished populations in the U.S. and abroad are neglected at times. I hope I answered your... Is there something else? Yes, please.
Hi, thank you for your talk, by the way. We appreciate it. There's an unfortunate link between malaria and sickle cell. So I was just wondering if throughout your travels in Africa, if you saw that link directly, and how did that influence your work, if it did at all?
Sure. The important first molecular disease, sickle cell anemia, Linus Pauling, 1949. We've known what causes sickle cell anemia for 75 years, but there's still no practical treatment. The CRISPR-Cas modification of stem cells will work, but at an enormous cost per individual patient. So it's quite interesting that the malaria belt doesn't include all African countries. And when we look at the individuals for the treatment of malaria, the areas where there's not endemic malaria, for example, southern Botswana, South Africa, the sickle cell hemoglobin is much less common. So it's quite clear that the sickle mutation, which erupted in at least 2 different locations, probably in Western Africa in ancient times, it spread because of the partial resistance to malaria. When everybody's sick, if someone's less sick, they'll have a survival advantage. And so that still is a factor, as well as the enzymopathies, which the glucose 6-phosphate deficiency, which affects which medicines you can use to treat malaria. So it's very much a factor. And sickle cell is historically the most important because it was the first molecular disease. And malaria, from my view, is the most important disease of red cells. And they fit together. Thank you. Okay, well, thank you so much.
So, greetings, everyone. We saw some impressive presentation today, and thank you all for being here at the Spring 2024 One Health Symposium And thank you, President Robbins, for joining us today. And by the way, I am Iman Hakim, the Dean of this wonderful Mellon Enoch Zuckerman College of Public Health. So we were honored to have Dr. Peter Agre from the Bloomberg School of Public Health at Johns Hopkins University with us today. Dr. Agre, as you know, is a Nobel laureate. He gave the keynote address in which he shared his insight on aquaporin water channel protein in humans and other organisms. This is fascinating and important work, so thank you so much, Dr. Agre, for joining us and sharing your expertise. Before Dr. Agre's keynote session, the engaging discourse on From Pixel to Public Health, Pioneering One Health Disease Surveillance by the team from the Ending Pandemics, opened our eye to innovative approaches to finding, verifying, and containing outbreak. Their dedication to shaping the future of disease surveillance aligns seamlessly with our collective pursuit of safeguarding global well-being. All of us who are here today and so many others across campus give our heartfelt thanks to Dr. Frank von Heppel for the outstanding work he has done to launch the One Health initiative here at the University of Arizona and the Health Science. He has brought together researchers from across the university and around the state to advance our understanding and expertise in the One Health arena. So thank you, Frank, very much. I also would like to mention and thank Dr. Mike Dick and Dan Dirksen for the support also they gave to the One Health initiative at the Health Science and the University of Arizona. So, and many thanks to Griselda Brown for her work, for Frank organizing today's event. Thank you, Griselda, for all the work. So as we move forward from this symposium, so let us continue to build on the connection and collaborations we have established here and work towards a shared vision of a healthier, more sustainable future for all. Thank you again for all the speakers and participants for making this year's One Health Symposium a success. Finally, thank you again, everyone. Thank you for participating, and let us collaborate and have a good evening. Thank you.